Chapter 6, Topic 2, Environmental Factors that Influence Microbes. In this topic, we're going to discuss an important term related to environmental factors. We're going to talk about what environmental factors influence microbial growth. We're going to talk about temperature, gases, pH, osmotic pressure, radiation, hydrostatic or atmospheric pressure, and how other organisms impact the environment. All of these things are going to help us understand how to, 1, grow microbes in the lab when we want to grow them, and 2, how to prevent their growth in areas where we don't want them growing. The learning objectives for this topic are to be able to name 5 types of bacteria based on their temperature preferences, be able to name how different organisms deal with oxygen, be able to name 3 physical factors besides temperature and oxygen requirements that microbes must contend with, be able to list and describe the 5 types of associations microbes can have with their hosts, and be able to discuss the characteristics of biofilms that differentiate them from planktonic or floating bacteria. Before we go any further, one important suffix that will help you make it through this topic is phile. This means loving and will refer to the prefix found with this term. For example, an aerophile loves air. Make sure you understand this because it will give you hints as to what the name of each microbe group means. Before we talk about how the environment impacts the growth and survival of microbes, we need to establish what environmental parameters they are sensitive to. Some microbes can survive in many extreme conditions, while others can be very sensitive to everything. The key to remember as we work through this topic is that each species of microbe is different and can be impacted by these in different ways. If I were to ask you about all the factors that contribute to the environment around you, I bet your list would look similar to this one. We will be talking about each of these on the following slides, but let's talk through what these are right now. Temperature is fairly basic. This can be hot or cold. We usually think of only hot when talking about microbes, but the cold can inhibit their growth as well. The next item is oxygen content. Because we humans require oxygen, it is easy for us to think that this parameter only deals with having too little oxygen. Some microbes grow only in areas with little to no oxygen, so in the case of microbes, there can be too much oxygen. We have talked about pH before, and just as temperature, each microbe has a unique pH range that it likes to live in. For example, the microbes in our stomach prefer a very acidic environment. The opposite is true for other microbes, and some, just like us, must be in a neutral pH. We talked about the importance of osmotic balance in the last topic, and you can see it here as well. It is important that microbes be in an environment that is consistent with their own needs. Radiation and atmospheric pressure are also important. Both of these can determine where microbes may be found, as well as how we can sterilize items to kill off any growth. If you think about the microbes that live at the bottom of ocean vents, these little guys can survive quite a bit of pressure, while our good friend E. coli won't survive as well. The last item on our list is why we love our microbiome. The presence of microbes can prevent others from growing in that area through production of antibiotics and other agents. Now that we have an overview, let's get started on each of these in more depth. Let's get started with temperature. There are going to be a variety of vocabulary terms used over the next 2 slides. I would recommend making some flashcards to help you get these down, because these need to become second nature to you. The first of these is cardinal temperatures. This is the range of temperatures a given organism can grow within. There are 3 sections within this range. Minimum is the absolute lowest temperature that this microbe can grow and perform the basic functions of life. Lower than this, and you get a microbe popsicle. That doesn't mean they always die. It can mean that they can hibernate if the other conditions are right. The maximum temperature is the exact opposite. This is the highest temperature a microbe can live at. While microbes have more flexibility on the lower end, if the temperature gets too high, they will die when their proteins start unfolding. Remember what this term is? That's right, denaturing. The optimum temperature is where you see the best and fastest rate of growth. Think back to the experiments you've been doing in lab this term. What temperature have you been incubating your assays at? It is likely the optimal temperature of the microbes you are trying to culture. We have divided bacteria into 5 groups based on their growing temperatures. You can see them indicated on the chart here. The microbes that grow and live in a refrigerator or freezer are known as psychrophiles. The microbes that have an optimum temperature around room temperature are known as psychrotrophs. Next are the bugs that we most often work with in the lab. They are mesophiles and tend to thrive at human body temperatures. On the other extremes are the thermophiles. These temperatures are bathwater and warmer temperatures. We then have the extreme thermophiles. These are organisms that we would find in extremely hot springs. What is important from this chart and information is that you understand these different groups and how the chart works. Make sure you notice how some of these little guys have a wider cardinal temperature range than others. Lastly, be able to use this chart to answer questions about a given bacteria. Let's move on to the second most important environmental factor, gases. As I mentioned in the earlier slide, oxygen is a major player in the growth demands of microbes. We have 5 main groups of microbes as far as oxygen demands go. The first of these are the aerobes. These little guys have oxygen demands like ours. They must have oxygen to survive and grow. Microaerophiles still need oxygen, but just not as much as those of us who need oxygen. They will be found growing in areas that have a limited access to oxygen, but still have it. The next are facultative anaerobes. These guys don't need oxygen, but if it's handy, they will use it. They are a very versatile group of microbes. It is very difficult to distinguish between facultative anaerobes and aerotolerant anaerobes. These little guys have a similar growth pattern to the facultative anaerobes, but don't actually use the oxygen. This is where their name comes from. They tolerate oxygen, but they don't use it. The easiest way to tell the difference is by looking at the difference in the growth. Aerotolerant anaerobes will typically have more sparse growth. Lastly, we have the anaerobes, sometimes called strict anaerobes. These guys do not grow in the presence of oxygen at all. We even have to use special oxygen-deficient chambers to culture these bacteria. Besides oxygen, some microbes need more carbon dioxide than is normally found in the atmosphere. These are called capnophiles. Don't worry about these carbon-loving bacteria too much. They aren't the main focus of our class. Our third environmental factor that impacts microbial growth is pH. While most organisms like you and I need a pH of between about 6 and 8, some organisms can only exist in very acidic or basic environments. Those microbes that live in acidic environments are known as acidophiles, while those that prefer a basic environment are known as alkalinophiles. In our last topic lecture, we discussed the importance of osmotic balance. Because of the importance of osmotic balance, most organisms need to live in environments where the outside environment is either isotonic or hypotonic. However, there are a group of microbes that prefer hypertonic solutions. Hypertonic compared to what we require, of course. These bacteria are known as osmophiles. It is important to recognize that this can be a variety of solutes, but we mostly talk about those that prefer high salt environments. These osmophiles are known as halophiles. There are 2 types of halophiles: obligate, meaning they have to have salt, and facultative, meaning it doesn't bother them. If you think back to how we used to preserve all of our food and what we still do today with canning, these halophiles can cause significant problems because the high salt concentrations meant to preserve food doesn't prevent these guys from ruining the party. Radiation is a common method we use in the laboratory and in industry to kill bacteria on a given surface. When preparing to work in a sterile environment in the lab, we use UV lights to sterilize the room. Some electronic toothbrushes even use this technology to kill bacteria on the toothbrush. Well, at least they say they did. We'll talk about that more on the topic of disinfection and sterilization, but the bristles actually block the UV light, and it doesn't even disinfect the bristles. Most bugs can be killed by enough of this radiation, just like your cells can be, but some can even be damaged by exposure to the radiation of sunlight, so they avoid the sunlight. Atmospheric pressure is important as well. Most microbes require regular atmospheric pressure, and by placing them in an area with a higher pressure, we are able to kill these microbes. This isn't true for all microbes. As I mentioned earlier, the microbes at the bottom of deep sea vents need significantly more pressure to survive and cannot live in the pressures that we exist in. These microbes are known as baryophiles. Think of barometric pressure. Besides all of these environmental factors, other organisms can impact the ability of a given microbe to grow in a certain area. This is why we love our microbiome. It helps prevent other microbes from growing through an antagonism action. Our microbiome is useful for another example here. Our relationship with the microbiome is a symbiotic relationship where both benefit. This is known as mutualism. Since we have spent this topic learning a million terms, I'm not going to go through all of these right now. You really only need to understand the basic concepts of these 5 types of relationships. Besides the antagonistic relationship our microbiome has with pathogens, there's another antagonism that comes from the phytoplankton in coral reefs. These organisms work to get the available nutrients in the area low so that pathogenic microbes, that is pathogenic to the coral, are unable to colonize the coral. Also on the side of synergistic relationships is the development of biofilms. Biofilms form a very cohesive community of organisms developing into a biofilm, and through their relationships they are able to succeed more as a community than they would on their own, much like a study group. Well, that's it for this topic. Be able to name the 5 types of bacteria based on their temperature preferences. Be able to explain how different organisms deal with oxygen. Be able to name 3 physical factors besides temperature and oxygen requirements the microbes must contend with. Be able to list and describe the 5 types of association microbes can have with their hosts, and be able to discuss the characteristics of biofilm that differentiate them from planktonic bacteria.